Hello, today we'll be talking about epistasis, a very important topic in understanding genetics, specifically inheritability, how an organism looks. It's called their phenotype, while their genetic makeup determines their genotype. Some traits are driven by one locus or set of alleles, while others consider multiple. Epistasis is the expression of alleles at one locus, or masks or modifies the expression of other alleles at another locus. When evaluating this topic, it's key to understand dihybrid crosses. A dihybrid cross is mating two homozygous organisms at two different loci. For example, two organisms of both big A, little a, big B, little b. When no epistasis occurs, meaning the result of one locus does not influence the other, you get a 9 to 3 to 3 to 1 ratio. Out of the 16 progeny, 9 show both dominant traits, 3 show the other dominant, and 1 shows only recessive. When epistasis occurs, this 9 to 3 to 3 to 1 ratio becomes modified based on the type of epistasis. Six main modes of epistasis: single recessive, single dominant, single duplicate recessive, duplicate dominant, duplicate genes with cumulative effect, and dominant recessive interactions. Each of these modes produce a unique ratio of interest. When a dihybrid cross is performed, these values are shown here. The mode of epistasis I will be covering today is single recessive epistasis. In this circumstance, when an organism possesses a single set of homozygous recessive traits, this will modify the expression of the other allele. This means that if an organism has two little a alleles or two little b alleles, they'll have the same phenotype. In our example, we're looking at the colors of dinosaurs, specifically looking at two different mating outcomes. In order to produce heterozygous dinosaurs for our dihybrid cross, we need to cross a purebred blue dino with a purebred red. The first cross is a parental generation produces a second generation F2 which consists of entirely gray dinosaurs, which are heterozygous for both A and B. Now we will cross two of the heterozygous gray dinos, which deserve the impact of epistasis. In this scenario, a ratio of 9 gray, 3 red, and 4 blue T-Rexes is produced. Any dino with a big A or big B will be gray. Any dino with a big A followed by two little B's will be red. Any dinos with two little A's or two little A's and two little B's are blue. There are only 3 phenotypes for our 4 genotypes. We will explore how this happens with revisiting our dihybrid cross. When you get the 4x4 Punnett square complete, it looks like this. You can see dinos with big A's and big B's are gray. Dinos with big A's and little B's are red. Dinos with little A's and big A's or little B's are blue. The key to this is understanding that two little A's produces a blue dino no matter what is at the B locus, as this is masked. As A is the locus that modifies the expression of B, this is the epistatic gene. Blue represents the starter. If you do not have a functional A enzyme, you cannot move on to the red intermediate. Now, if the red intermediate is reached, dinos with big B's are moved on to the gray color, while those with little B's remain red. Now we're looking at an example where this epistasis does occur, though not in a dihybrid cross. As a result, the 9 to 3 to 4 previous ratio in previous dimension is no longer valid. In this case, we're looking at big A, big B mating with a, little a, big B, little b. This will be a gray dino crossing with a blue one. When you perform a 4x4 Punnett square, you observe much different result than before. As A remains epistatic to B, the 8 results that start with two little A's are blue while the rest are gray. This 8 to 8 split produces a 1-to-1 ratio. The key is that the cross is not a dihybrid and A is epistatic to B.